Welcome back. Today is August 20, 2026, and we're diving deep into something that honestly feels like science fiction, but it is happening right now in the medical world.
Yeah, it really is wild.
You are the ultimate learner, and you want to get past the heavy jargon to uncover the real aha moments in modern science, right? Well, today we are going to do exactly that. We've got a really fascinating stack of sources for today's mission.
We do. We're looking at a great collection of immunology reviews, some cutting-edge biotech patents, and some really surprising lupus research.
Yeah, the lupus connection blew my mind.
Mm.
But our mission today is to answer a highly specific but completely revolutionary medical question. We want to know, how does the removal of double-stranded DNA immunogenic epitopes prevent human anti-DNA antibody generation during polynucleotide therapy?
I know. It is a massive mouthful.
It is. But okay, let's unpack this.
Yeah.
Because DNA therapies, things like advanced vaccines or gene therapy, they are incredible.
Right. They have so much potential.
But they face this massive hurdle. Our own bodies often attack the cure. We inject a life-saving DNA sequence, and the immune system just destroys it.
Exactly. And my role here is to kind of bridge that gap. To make these DNA therapies actually work, scientists have had to figure out, exactly what sets off the body's alarm system in the first place.
Right.
And then once we know the alarms, we have to figure out how to systematically strip those alarms away from the therapy.
So to understand how to remove those alarms, I guess we first have to understand what the alarms are, right? Like, why does the body even care?
Well, it comes down to how traditional polynucleotide therapies are made. Historically, we've used something called plasmid DNA.
Okay, plasmid.
Yeah, plasmid is basically just a circular loop of DNA, but it has a bacterial backbone. We use bacteria to grow it in the lab.
Right.
And our innate immune system, it uses these things called pattern recognition receptors, or PRRs. They are constantly looking for pathogen-associated molecular patterns, or PMPs.
Wait, so when we inject a traditional DNA therapy, the body is sensing that it came from bacteria. I mean, how does it even know? Isn't DNA just DNA?
You'd think so, but no. Bacterial DNA is absolutely full of these unmethylated CpG dinucleotide motifs.
Unmethylated CpG motifs. Got it.
Basically, it's a specific spot where a cytosine sits next to a guanine. And mammalian DNA, like ours, it has very few of those.
Okay.
And when we do have them, they are usually highly methylated.
Highly methylated, meaning like they have a chemical tag on them.
Exactly. A little methyl group is attached. It's kind of like a molecular do not disturb sign. It tells the immune system, hey, I belong here. But bacteria don't have those tags.
Oh, wow. So the unmethylated bacterial DNA just screams foreign invader?
It does. When it enters a cell, intracellular receptors, like one called TLR9, they detect those raw CpG sequences and just freak out.
Right.
And then it gets even more intense. There's another sensor called the STING pathway.
Sting. That sounds appropriately aggressive.
Yeah, it stands for stimulator of interferon genes, and S-STING binds directly to cytosolic double-stranded DNA. Doesn't even need any accessory molecules.
He just grabs it right out of the cell fluid.
Exactly, straight out of the cytosol. And once it binds, it activates an enzyme called TBK1.
Okay, TBK1.
Yeah, tank binding kinase 1. STING basically escorts TBK1 to these endosomal compartments, which activates transcription factors, things like IRF3, IRF7, and NFKB.
And those transcription factors, they trigger the actual immune attack.
They do. They blast the body with pro-inflammatory cytokines and type I interferons. It's a massive inflammatory response.
Okay, so it's like mailing someone a beautiful life-saving blueprint, but you've stuffed it inside an envelope that screams, I am a bacteria.
That is exactly what it is. The body burns the envelope before it can even read the blueprint.
But wait, I have to push back here. Our own bodies are literally full of double-stranded DNA, right?
Right, trillions of cells.
So how does the immune system tell the difference, just based on shape or these CpG markers, without accidentally attacking ourselves? Like, cells burst all the time.
That is the million-dollar question.
Right.
And to figure it out, immunologists had to look at a situation where the body does attack itself.
Oh, autoimmune diseases.
What's fascinating here is that the clues came from systemic lupus erythematosus. Lupus.
Okay, so how does lupus help us understand DNA alarms?
Well, in lupus, patients develop these anti-dsDNA antibodies. They deposit in the kidneys and cause lupus nephritis.
Which is really severe.
Extremely. But the paradox in the research was that Classic right-handed double helical bDNA, the standard Watson-Crick DNA.
The normal double helix shape.
Yeah,
Yeah.
it completely fails to induce autoantibody production in animals in the lab.
Wait, really? The immune system just ignores normal bDNA?
It completely ignores it. The immune system doesn't target normal bDNA. It targets unique structural epitopes.
Structural epitopes, meaning weird physical shapes.
Exactly. Specifically, something called zDNA.
zDNA. Okay, if bDNA is the normal right-handed staircase, what is zDNA?
It is an energetically unfavorable left-handed zigzag helix.
Well, left-handed zigzag.
Yeah, it is a very unnatural shape. It forms under specific stress conditions like heavy base methylation or chemical modification. For instance, brominated poly-DGDC.
So basically deformed DNA.
It is. And here is where it gets crazy. The microbiome connection. Bacterial biofilms, like those from Salmonella or even commensal gut bacteria, they can produce factors that actually trigger these specific anti-DNA antibodies.
Oh, I see. So the immune system isn't just wildly attacking all DNA. It's actually primed to generate these anti-DNA antibodies only when it detects specific structural epitopes,
Right.
like the zDNA zigzag or chemical markers like those unmethylated CpGs.
Exactly. It is highly specific to the shape in the chemical tags.
Okay, well, here's where it gets really interesting, because is specific structural epitopes in bacterial CPK backbones are what trigger the alarms? Couldn't we just physically remove those triggers from our therapies?
Just trim the fat. Yes, absolutely. And that is
Yeah.
exactly what the new generation of DNA vectors is doing.
Right. The sources talked about a few of these: minicircle DNA, doggy bone DNA, and hairpin DNA. Let's start with minicircles or MISI DNA.
Yeah.
How do they work?
So with minicircle DNA, engineers take the original plasmid and just cut out the bacterial backbone entirely.
Just slice it right out.
Yep. They remove the antibiotic resistance genes and all those CpG-rich sequences, leaving only the eukaryotic expression cassette.
And by removing the CpG sequences, you prevent that TLR9 activation we talked about.
Exactly. And it also stops epigenetic silencing.
Epigenetic silencing, what's that?
It's when the cell slowly turns off a gene therapy over time because it realizes it's foreign. But without the bacterial signals, the therapy can keep working long-term.
That is so clever. Okay, what about doggy bone DNA or dbDNA?
Doggy bone DNA is great because it is a closed linear format that is actually made with cell-free enzymes.
Oh, so they don't even use bacteria to manufacture it.
None at all. It's entirely synthetic, which completely eliminates the risk of bacterial CpG contamination.
Wow. And the third one was hairpin DNA, HPDNA.
Yeah, hairpin DNA is similar, but it's covalently closed by single-stranded hairpin ends.
Like the ends of the DNA strand fold back on themselves.
Exactly. It protects the DNA from getting chewed up by enzymes while still stripping away all those immunogenic epitopes. It prevents the immune-mediated clearance of the transduced cells.
Okay, so we strip off the I am a bacteria envelope and just deliver the blueprint.
Yeah.
But, hold on. Even if we use minicircles or doggy bones, it's still double-stranded DNA, right?
It is.
And you said earlier that the STING pathway specifically hunts for double-stranded DNA in the cytosol. So wouldn't just the sheer presence of a double helix still trigger STING?
You've hit on the absolute cutting-edge frontier of the field.
Yes.
For some highly sensitive applications, any double-stranded DNA is still too risky.
It's like sneaking past a security guard who is specifically trained to look for pairs of intruders. If you walk in entirely alone as a single strand, you don't even register on their radar.
That is a perfect analogy, and that brings us to the ultimate disguise. Single-stranded DNA, or ssDNA.
Wow, so they just drop half the ladder.
Basically, yeah. A recent patent on non-viral ssDNA expression systems shows that ssDNA is a significantly less potent activator of the CGS STING pathway.
Because it just doesn't physically fit into the STING receptor.
Exactly. It's too thin and flexible. STING wants that rigid double helix. So ssDNA completely bypasses those innate immune sensors.
That is brilliant.
And clinically, this is huge because it drastically expands the therapeutic window.
Oh, right. The therapeutic window.
Right.
That's the dosage range between an effective dose and a toxic one, right?
Yes. If the immune system isn't attacking the therapy, you don't have to use massive toxic doses just to get a little bit to work. You can use a much safer dose.
And that ties right into this delivery mechanism that Velvet is working on, right?
Right.
Their in vivo CAR-RT platform.
Yeah, Velvet's tech is the pinnacle of this stealth delivery. CAR-RT therapy normally requires taking cells out of the body, which is exhausting and expensive. Velvet wants to do it in vivo inside the body.
So how do they get the single-stranded DNA in there without triggering an alarm?
They use a polyplex delivery system, specifically utilizing cationic polyaspartic acid polymers.
Cationic.
Yeah.
Meaning positively charged, right?
Exactly. DNA has a highly negative charge because of its phosphate backbone.
Oh, I see. So opposites attract.
Right. The cationic polymers neutralize the negative charge of the DNA, and it compacts the whole thing down into these incredibly tiny nanoparticles.
It basically shrink wraps the genetic material.
It really does. And then these tiny positively charged nanoparticles interact with the cell membrane, and they enter the cell via clathrin-mediated endocytosis.
Clathrin-mediated endocytosis, so the cell just naturally swallows it.
Basically, yes, it completely evades immune detection. And the most amazing part from the sources is that it can express its life-saving instructions, like building a scar against solid tumors, in as little as 2 hours.
Wait, really? 2 hours?
2 hours, all without triggering any anti-DNA antibody response.
That is staggering.
Yeah.
So, what does this all mean for you, the listener? Let's trace this back. We started by looking at how diseases like lupus generate anti-DNA antibodies by targeting specific things like CpG motifs and zDNA structures.
Right, the immune system reads shapes, not just the concept of DNA.
Exactly. And by understanding that, scientists figured out how to engineer polynucleotide therapies that physically strip away those exact double-stranded and chemical epitopes.
It's incredible engineering.
Yeah, whether it's through minicircles, hairpins, or just fully single-stranded DNA, we can now actually rewrite cellular code without setting off the body's alarms.
And this is why this fundamental knowledge matters so much. Understanding the nuances of molecular structure, like the zigzag of a left-handed helix, is now paving the way for off-the-shelf in vivo CAR-RT therapies.
Therapies that could actually cure cancers without those devastating immune side effects.
Exactly. It's revolutionizing medicine.
It truly is. But before we wrap up, there is one final kind of provocative thought I want to leave you with.
Oh, okay. Let's hear it.
So, it was buried in the margins of the source material. We noted that anti-dsDNA antibodies in lupus are heavily linked to immunity against commensal gut bacteria, right? Things like Plautian novis or Salmonella.
Biofilm producers, yeah.
Right. So if our immune system's baseline hostility to DNA is actually trained and calibrated by the bacteria living in our gut, could a patient's daily diet or even their probiotic intake ultimately dictate whether a life-saving DNA therapy is accepted or rejected by their body?
Oh, wow. That is a wild implication.
I mean, are the bacteria in your stomach secretly controlling your genetic future?
It really makes you think about how interconnected everything in the body is.
It really does. Definitely something for you to ponder next time you take a probiotic or drink a kombucha. Thank you so much for joining us on this deep dive. Keep questioning, keep learning, and we'll catch you next time.